Thanks to Teresa and all the Fidelio team for inviting me to share this webinar today. I hope that all the participants will find it interesting, and please, if you have questions either during the webinar or maybe if you want to contact me through mail, do not hesitate and we can discuss further.
Carolina, on that point, let me add, if you have questions, please submit them through the chat. I will be reading them, and at the end, we will have time to look at the questions.
Okay. So it is already some years since we have noticed that we are more than humans. We are what is known as holobionts, and this means that we are a whole ecosystem. And why do we say that? We say that exactly because we have noticed that actually have more bacterial cells than actually human cells that are able to process and carry out metabolic processes in our body. So if we look at the comparison between the number of human cells and microbiota cells, the ratio is 1 to 1.3. At the same time, if we will make a comparison about the number of genes that are present, that are able to carry out functions in our body, it will be 1 to 100 on the bacterial genes. And this is like the more stringent ratio. So if we are talking about 23,000 genes that are coded in our genome, actually, the people that is less optimistic about the gut microbiome, they think that just in the gut microbiome, we should be having between 5 million and 23 million genes that are able to carry out metabolic processes. So with this, I guess for you, it's not surprising then that I tell you that there are many essential functions on the human body that actually are not carried out by our human cells, but by the bacterial cells. So most of the vitamin K and vitamin B that we actually have in our body comes from the metabolism of those bacterials. Also, we know that short-chain fatty acids, and this is something that you will see throughout the presentation. So SCFAs actually are the product of fermentation of some of those bacteria in our gut. And these molecules are really important as they are able to really put in function many signaling pathways in our body and chain reactions. So also the microbiome is able to transform the bile acids, and there are many of those compounds that are metabolically important, and that if you don't have the microbes that are actually able to do those processes, then it means that you will lack those biotransformations. And indole is another important molecule that we know is just a subproduct of the tryptophan pathway that is carried out by our microbiota. So as it is expected then, give me a sec. So as it is expected then, the gut microbiome has been discovered, and I say discover just with quotes because it was already there, but now we discover it as an integral component, a component of our health. And what we can imagine is that the microbiome will play a role in nutrients bioavailability, immune modulation, pathogen protection, dysregulation of cellular cycles, and as such will then be associated with an enormous number of traits and diseases, and between those, the ones that perhaps have called more the attention of the scientific community are the metabolic syndrome, the type 2 diabetes, cardiovascular disease, Alzheimer's disease, and all type of cancers, but not just cancer, but respond to treatment of cancer patients. Nevertheless, the things are not as simple as it looks like, because there are a lot of feedback mechanisms and loops that are carrying out when we talk about the microbiome. So you can imagine that the microbiome is highly affected by the host genetics, by the environment, by the nutrition, by the lifestyle, right? So depending on what you eat, your microbiome will take one shape or another shape, and those shapes then will result in some sort of metabolic changes that perhaps will then influence your risk to get a disease. But at the same time, perhaps the fact that you get certain disease will alter your diet again. So it's all the time like this vicious cycle. So it's really hard to disentangle what is first. Is the microbiome the one that is changing, and as it is changing, then there are some consequences that at the same time will produce a change in your diet, or is it the other way around? And you can think in that way for almost every variable that come to mind. In the sense, in the field of bone biology, that perhaps is the one that brings us together today, right? There are so many studies that have been emerging in the last years, that actually we have one new word, that is osteomicrobiology, in which we are defining or trying to frame all these studies together. So the new term osteomicrobiology for the rapidly emerging research field on the role of microbiota on bone health. This research field is aimed to bridge the gaps between bone physiology, gastroenterology, immunology, and microbiology. And actually, it is the focus of the webinar that I will be presenting today. So the effects of the gut microbiome in bone metabolism are clear, they exist, and although we don't have a clear vision exactly how they happen, there are many different pathways in which the gut microbiome has been implied, and as such, then we think that those pathways will be the actual mechanism underlying those associations that we see between the gut microbiome and bone. So the first one is that the gut microbiome is able to regulate the nutrient absorption from the diet. So in the case of bone, perhaps the most important will be how we are able to absorb some minerals as calcium, phosphorus, or magnesium, and how that absorption then will result in different outcomes on bone traits. We also know that the microbiome is able to affect the translocation of microbial products across the gut endothelium. So imagine that the gut is an ecosystem, and that permeability between the gut lumen and the rest of your body can change, and it will change depending on what is there on the level of inflammation. And if it is leaking, so if it is more permeable and more things can cross through it, you can imagine that that will also generate different responses in our body. Also, we know that the microbiome is implicatedly really strong on the immune modulation. So it not only when you are a baby will be the first, let's say, antigens that will be shown to your immune system, so that you develop all these defenses, but also through life is one of the factors that will play a key role on the activation of CD4 plus T cells, meaning that it will directly affect osteoclastogenesis. And of course, it has been proved that also it is not only implicated on this bioproduction of vitamins, but also it will affect the levels of sex hormones or serotonin in the body. So how I first thought about this presentation is that I will go just through a timeline and tell you what we know and more or less how we discovered it and in which order it was happening. But then I noticed that perhaps it is easier to move from topic to topic and talk about all the studies that have, let's say, support the same conclusion, and make more an emphasis on what we know from basic science from mice models and what we know actually from human studies. So let's see how it goes. I hope that is clear, and please feel free to put your questions on the chat, and we will then discuss it at the end. So the first thing that we will consider, as I mentioned, is that we will start by the mouse models, and then it's important that you keep in mind some important terms, terminology. So for example, when we talk about germ-free mice, we are talking at those mice that actually they are already born on sterilized cages. So they have never, and they will never during the experiment, have any kind of contact with bacterial material, in opposition to conventional raised mice that are just the ones that are raised on the lab. Just giving normal food and having contact with their own faces and all this. And also there are the ecnotobiotic mice. It means those mice that if they are germ-free, then you will give them some bacteria that you will feed them, and you will know exactly what is there, right? So I can put a specific bacteria on their diet, and I know that the only bacteria that they have circulating in their body is actually what I gave. And this is also the principle for those humanized mice that we call, and those are the ones in which I will just give them encapsulated the faces of some human participant, either a person that have a particular disease or even just a person in the population, to make a comparison on the phenotypes that will emerge on the mice. So I will start by the first study. Of course, in the past, we already knew that the microbiome perhaps was playing a role in bone, since from all the previous work in agriculture and cattle and all this animal raising for our alimentation, we knew that probiotics and prebiotics will play a role on the development of muscles and bones on these animals, right? So the first studies that are actually trying to assess in mice models what is the consequence of having a microbiota versus not having it were published around 2012, and they mostly come from the group from Klaas Olsson and Clara Sjörgren in Gothenburg. So this, I think, is the first paper that they did, and as you can imagine, at that time, they are really simple analysis in the sense that I am not looking really to particular microbes that I am interested on. I am just raising these germ-free mice, and I am just raising these conventional mice, and I will compare at the end of the day, I don't know, in this experiment at 9 weeks, how do the bones look from one group and how do the bones look in the other group. So what they actually saw was that the germ-free mice, they will have higher BMD, and they will have higher cortical bone area, among other parameters. Of course, I didn't put all the figures that they have in their paper. But let's say that in general, the germ-free mice seem to have a much better bone parameters than the conventional raised mice. And in this case, perhaps you are wondering then what is this third group? And this third group is simply that after 3 weeks, they took some of the germ-free mice and they gave them some phases from the conventional raised mice to try to mimic so that the microbiota that was in the conventional raised mice will engraft on their gut of these germ-free mice and see what are the consequences on that. So they see that they are closer to the conventional raised mice than to the outcomes on the germ-free mice. So when they saw these phenotypic changes, then they started to look at the cellular level what was going on. Why should the germ-free mice present with such a better outcomes in comparison to the conventional raised mice? And they saw that actually the germ-free mice had much less of these CD40 cells, meaning those that will give rise to the osteoclast. They have much less pro-inflammatory cytokines. And of course, as I mentioned, there were less osteoclast precursors. So as we are all in this bone field, we can imagine that if the osteoclast activity is less, then we will have less bone loss. And then at the end of the day, we expect to see a higher bone mineral density. Actually, those results were then replicated in several studies in which they did practically the same kind of analysis. So they took these mice. I make a lot of emphasis to actually which strain of mice they use in all the different experiments, and at what time did they do the inoculation of the microbiota, because as we will see later, those seem to be really important variables when you try to actually look at all the results jointly. So the same group of class also, they repeated the experiments, but at this time, for example, they use mice from 2 different institutions, because there is a lot of criticism when the studies carried out just in one institution because of the genetic background of the mice, and they saw exactly the same. So all these markers that we see here, like TNF alpha, rank L, and Katepskin K, we know that they are all markers from osteoclastogenesis, and as such, what they are saying is, yeah, in conventional raised mice, this is much higher, but from the germ-free mice. This was also observed by another group in which they look actually to the consequences of estrogen depletion in overectomized mice. So they look at how it looks for a mouse that actually will have this estrogen depletion, but at the same time was germ-free mice, and how it looked for conventional raised mice. And they saw actually that for the germ-free mice, it seems that the consequences, the bone loss after this estrogen depletion, is not really strong, whereas for the conventional raised mice, it is the change in bone really striking. And also, there was another paper in which they look exactly at the same. So they have the SPF mice, and SPFs just mean that it is without pathogens. So they are conventional raised, but they make sure that there are no pathogens anywhere on these mice, not to have that bias. But so everything up to now seemed to be really nice, in the sense that there are a lot of support for the hypothesis that actually germ-free mice will have better bone outcomes, so that somehow the microbiome, the microbiota itself, is detrimental to bone outcomes. But there are some conflicting results also from the same time in literature, in which they look either to other mouse strains like Swiss Webster, or they look, or bulb C mice, or they look also to different designs. So when it was colonized, when the measurement was done, and you see that in this case, for example, Quash et al. Showed totally that there is no significant difference between the germ-free mice and the conventional raised mice. So they don't see the same effects that until now all the other studies were reporting. Whereas Swasher et al. In Science, they say, hey, look, when we look in these bulb C male mice, what we see is that actually the wild type, so the conventional raised mice, have longer bones, right? So there are some conflict also on the findings, that you have to see everything on the light of what factors can be playing a role on getting those conflicting results. And the first one is, of course, that when we are talking about two different mice, perhaps there are a lot of differences, and there are a lot of papers that have claimed a lot of difference, especially on the immune development of the different strain of mice that will have for sure consequences on the bone traits. Also, many of the first studies, well, as with many other outcomes, are done on just mice that are females. There is also a difference on the age. So what we know from other type of studies, not only contemplating bone, is that perhaps it has an advantage that you are an early conqueror, that as the microbiota, like that microbiota that perhaps reach the babies at the beginning, will define a lot of the environment that there is in the gut, and as such, it will somehow shape who can actually enter to that ecosystem and who is left behind, right? The age of inoculation then will play a role. So it seems that when it is early inoculation, then first of all, it is easier that those microbiota actually engraft on the gut, that they will actually stay there, and also that they will be more successful even when other microbiota will enter into the gut. The type of inoculation, so it is different if I have the germ-free mice, right? Or if, for example, I have just conventional raised mice, and then I have a period in which I give them antibiotics, like really strong antibiotics for a period of time. And then I say, okay, now these mice are also free from any microbiota because I kill everything that was there. And then I will put new microbiota, so we know that those antibiotic treated mice are different than the germ-free mice, right? And this was actually shown by a paper in PNAS on 2016, in which they were looking, for example, what happened with the inoculation of microbiota on these mice, what is the outcome on the bones, and they look after 1 month of the colonization, and in there they see those differences that were described on those early papers, that the germ-free mice will have better outcomes for bone traits. But then if the colonization already took 2 months, then they say, hey, look, we don't see differences anymore. So it seems that all those small variables are playing really a role, and that perhaps they explain why we have so many conflicting results in literature. As I mentioned already, not just density was one of the outcomes that was looked at, but also bone growth. So how can the gut microbiome affect bone growth? So what we know and what has been proved is that actually these short-chain fatty acids that are acetate, propionate, and butyrate, among others, are able to leak out of the intestine, go on your bloodstream, and somehow they will reach the liver, and they will say to the liver, hey, produce a little bit more of IGF-1. And then that IGF-1 is one of those key factors for the longitudinal growth of bone. So the microbiota is able to signal to produce more IGF-1, and as such, that there are longer bones as a consequence. And this is one of the earliest paper that they actually published. And what they saw also, it was that they could mimic the same effect just by giving the mice Lactobacillus plantarum WGL. And in here, I just want to call your attention that I'm not saying giving them Lactobacillus. I am saying Lactobacillus plantarum WGL, because it's really important for us to take into account that even every species can have a lot of strains, and those strains can have specific effects on bone traits. So in here, for example, if you look at the figure that is on the right, you can see my cursor? Fernando?
Yes.
You see my cursor?
No, not. Yes,
no?
Now it's here.
Okay.
It's a little bit small, but it's okay. You can see it.
Okay, so if we look at this figure and we see what we have in red and in blue, you see that the outcomes are actually significantly different. So it seems that actually giving the mice this Lactobacillus plantarum, that is another strain, the strain NISO 2877, do not have the same consequences and giving them the Lactobacillus plantarum WJL. So this is just a different strain, and as we see, the consequences are pretty different on the bone outcomes. Also important is that since 2018, there has been, I just wrote one publication, but if you look in literature, there are at least 3 or 4 more publications in which they are studying this Irdoviridae family, that is a family of viruses. These viruses also living in your gut. And it seems that the family of this virus is able to produce some peptides that look really alike IGF-1, and that actually they look so alike that they are able to fool the receptors and actually activate the same pathways that IGF-1 will be able to activate. So in this sense, then, as this Irdoviridae family is also one gut virus, then also the levels of this LCDB1 that are produced by this family could be one of the reasons that we have an effect of the gut microbiome, that of course we call the gut microbiome, and we talk all the time just about bacteria, but as you can imagine, there are also fungi and there are also viruses there. So, as I mentioned at the beginning of my presentation, another of the hypotheses of how these effects of the gut microbiome on bone is happening is actually the absorption of some of the minerals that are important. And in this case, I just brought some examples of the studies that have been carried out regarding calcium absorption on the intestine. So what we see here are studies like in rats, in which they give this gauze is just fibers that they give to be able to, that the bacteria in your gut are able to actually fermentate those fibers and then produce other things. And when they studied this in rats, they saw actually that the calcium absorption was increased on the bones, and that the volumetric BMD at the distal femur also increased on those rats that were given these fibers in opposition to the ones that were having just a normal diet. Also, when they look in detail to what changes on the microbiome were taking place, they saw that the bifidobacterium were increased in those rats that were given gauze in their diet. Another study that was actually made on 12 postmenopausal women, it was just to give them also this kind of fibers, not necessarily goats, I think it was almidon fibers, yeah, corn fiber, soluble corn fiber, that they give in the diet, and they look at how that changed the bone calcium retention, and they saw really an increase on that. In here is also the same type of studies in which they are looking at which changes actually happen in different taxonomies of the gut when you change the diet and give them something to be fermented. So most of the studies up to date, they have focused on the effect in bone resorption. So for this type of studies, it is always nice to have a mouse model in which you have estrogen deficiency. So they try to mimic what happened to a woman when a woman has menopause. And they do that by overectomize the mice, either chemically or by having a surgery. So what are the consequences of that estrogen deficiency in the gut is what I will explain really fast in here. So we know, first of all, that when you have this estrogen deficiency, the number of Th17 cells increase, and these Th17 cells just means that they are the precursors from osteoclasts, so that the number of osteoclasts at the end of the day will also increase. The pro-inflammatory cytokines also increase at the same time, rank L increase, and as I explained earlier, and really importantly, the gut permeability also increase. So, and with this, what I mean is that many of those products from the bacteria that perhaps in a person, in a young person, are staying inside the intestine, they will go outside the intestine to your bloodstream, and in there they will activate different type of pathways. What we know is that many of them, they will activate inflammatory pathways, and the levels of inflammation, high levels of inflammation are associated with worst bone outcomes. So this is the study that I mentioned before. So in this case, for me, it's really a landmark study that they did. Is really well done, but basically is just to look at the consequences of overectomized mouse, what happened to their bone. So this is how it looked before for the conventional raised mouse, in opposition how it looked when you overoptimize this mouse. So you see that there is a lot of bone loss on the trabecular, especially. I mean, more clearly on the picture at least. But actually, if those were germ-free mice, even if you overoptimize the mice, there are not consequences, at least at a significant level. Perhaps you see changes, but they are not significant. So what we see is really small changes in comparison with the conventional raised mice. And then they also look at what happened with those to which I will also give probiotics or other things, or the colonized, in this case, colonized germ-free mice. Yeah, so these were germ-free, but then at a certain point, 3 weeks after they were born, I start feeding them with the feces from the conventional raised mice to try to look at how it looks like to have the gut microbiota from a conventional raised mouse. And then I compare also what happened if I overoptimize some of them, right? So in terms of TNF alpha, Rankel, and EL17A, we see what we expected after looking at these figures, and is that for the over-optimized mice, we will see an increase in TNF alpha, Rankel, and those pro-inflammatory cytokines, whereas when we have the germ-free mice, those changes seem much smaller and even not significant between the over-optimized or the normal mice, as you can see in the middle of the different three panels. So also Su et al. In 2017, they show that, yeah, this is happening, and we know. But actually, if you will give probiotics to this mouse, you are able to revert the effect of the estrogen deficiency. So my take from all these studies is that girls, when we go into premenopause, it's really important that we take a lot of care from our gut microbiota, and then it will be a nice time to start taking probiotics if you are not taking right now. And in here, I just mentioned some of the studies, as I mentioned already, for osteoclastogenesis, for the effect of microbiota on bone loss, on osteoclastogenesis and bone loss. There are many studies, and in here I show the different studies, which kind of probiotics they have used, and also in red you can see the ones that have actually go beyond animal studies, because also there are clinical trials in which they have used these different probiotics and show the effect of those in bone. But I will discuss that later. So now I think for everyone it's clear that there is an effect of the gut microbiota on bone, that effect, at least for some time period and in mouse models up to now, we are able to see effects on bone just by giving probiotics. So by probiotics, I mean to give a specific kind of bacteria, and you give it live. So you give it in capsules, and it's supposed to then go through your system without being damaged, those capsules, until it reaches your intestine, and then they can live and reproduce there. So here is another study with another kind of murine model, and this is really an old rat, 26 weeks. And what they show actually is that when you give this type of probiotic, you are able to increase the osteoblast surface in the bone, to decrease the osteoclast surface, to increase the number of osteoids that are seen on the isomorphometric measurement at the femur. And here they are comparing those results graphically between the sham. It means those rats that actually didn't have a treatment, those that had a treatment, they were over-optimized, and those that were over-optimized, but they were giving this probiotic. And what we can see is that it is almost rescue. Some of the parameters are almost rescue at the end of the day, or at least improved when you just give the probiotic. And although most of our knowledge is how the gut microbiome can affect osteoclastogenesis, there has been few articles that have also tried to address how the gut microbiota can affect osteoclastogenesis. And here I brought two examples, one published in 2017 and another in 2018, with two different kind of mice, and actually they are just looking at the number of cells, how much RANX is actually expressed on the osteoblast from these mice that are germ-free versus those that are conventional raised but without pathogens. And the same kind of approach was used by Uchida et al. In Japan, and they also look at different molecules as OPG, rank L, osteocalcin, and ALP. And what they see is that the better outcomes that they see for the germ-free mice at the level of the bone and everything, they seem that are really related to the fact that on the conventional raised mice, on the ones that have microbiota, there is larger level of osteocalcin circulating. This is a really nice study that I just discovered last week when I was preparing for this webinar. It was actually published in 2021. And what they're showing here is that actually just the child, the gut microbiome that comes from children was able to rescue all the phenotypes in the overoptimized mice in opposition to the ones that were coming from the elderly population. So the elderly gut microbiome was not able to rescue, and the consequences of overoptimization were exactly the same as for the sham. Whereas when they encapsulated this child gut microbiome, they were able to see positive changes in many bone parameters. So I really thought it is really well written and everything, so I really recommend it for you. And then what they did was to compare like what happened with the profiles of the mouse like that have the same age. Then they overoptimize them, and then they see what changes happen before the overoptimization and after. So when there is this estrogen depletion, how does the microbiota change? And they look at those changes and they say, look, before we had this yellow, so the first three columns, we had this yellow, and then now we don't have it anymore. And that yellow is Akkermansia. And then they say, oh, but look, when we gave those rats, those mouse, the child gut microbiota, the Akkermansia came back. So it's here. But actually, if you give them the gut microbiota from the elderly, there is not Akkermansia coming up. So they thought, perhaps this Akkermansia is really important for all those good outcomes that we are seeing on the bone after giving this child gut microbiome. So what happens if I give them actually this Akkermansia by itself? And this is what you are seeing here. So it seems that Akkermansia itself is able to produce BMD changes. And the other things that you are seeing here is because Akkermansia is able to secrete some vesicles, and they think that those vesicles are the ones that are carrying out something outside the intestine, and that's something that they are carrying out is what is activating or deactivating inflammation and all that, and that's why we see that positive effect on bone. So they also took those Akkermansia that they want to give to the mouse, and they inhibit the possibility from those Akkermansia to generate these extracellular vesicles and see what is the effect. And they saw actually that they were right. They were right in which, in the sense that it seems that if the Akkermansia or the child gut microbiome is giving this treatment to inhibit the generation of extravesical cells, then it seems that then nothing happened, that the rescue of the phenotype is not actually achieved. But not only we have seen in literature changes in bone mineral density or in bone growth, but also there are studies that focused on seeing changes in bone strength. And actually, as you can imagine, the bone strength doesn't necessarily couple with bone mass, in the sense that it can be that two persons have a similar bone mass, but depending on how it is distributed on the bone, then they will be more susceptible or less susceptible to fracture. And that's why we know that the microarchitecture, the architecture of the bones matter, how the bone is distributed. But you can imagine also that there are other issues, like how the collagen is linked, or if the matrix is homogeneous or is heterogeneous, and all those factors will play a role at the time to define bone strength. And for this analysis that I mentioned here, they look actually to those properties of bones, or how heterogeneous or how homogeneous was actually the material itself, and they saw changes on this valve C mice at 16 weeks after treatment with Lactobacillus acidophilus. So they saw that the crystallinity was increased when you have added this Lactobacillus acidophilus to the diet of the overoptimized mice, and then you will see that the crystallinity will decrease. That's what you expect, right? So you expect that if the crystallinity is lower than the bone is strength, the properties of the bone. Also, they look at the mineral to matrix ratio. So what we know is that that mineral matrix ratio is better if it's low, meaning that the bone is a little bit more flexible. It's not that susceptible to cracks. And also they look at the carbonate phosphate ratio, and they saw differences. So all these just point out to the fact that it seems that the changes that the microbiome is able to produce on bones, they are really to the cellular level, to the micro level, right? So we see changes on bone mineral density, we see changes on growth, on longitudinal growth, but we also see changes on the properties of the material.
Carolina, just a reminder, we are approaching the time for the questions.
Thank you, Fred. So this is another study that was published in JVMR 2021. And in this study is the same principle. They look also, so this I think is super smart. I never thought about this, but that if you give different courses of antibiotics, then you are selecting different type of bacteria that will survive those antibiotics, right? So that's how they make the comparison. So they didn't make the comparison by group. They just took different groups and they give them different types of antibiotics, and they saw then how all the properties of the bone changed based on that. But of course, if you compare the profiles, you see some differences that are clear there.
Hey, ja, even aan. Ik had wel jullie
link. Okay. So I have gone through all the studies that we have right now, not all of them, but let's say the ones that are touching in different mechanisms on how the gut microbiota can affect bone. But it is important for you to know that I mentioned just the direct effects on bone, but you can imagine that if the gut microbiota, as it has been shown, can, for example, also influence muscle mass or muscle composition, you can imagine that that also can influence bone itself. So from all these mouse studies, we have learned a lot. We have a lot of principles, although nowadays there is a lot of controversy on the gut microbiota. There are a lot of papers trying to say what is wrong about all these mice models, and let's say what they say, well, actually the mice, as with every type of study, the mice is not a human, it's just a model. But importantly, there are really a small amount of bacteria that are able to engraft on the gut of a human that will also engraft on the intestine of a mouse, because they have a different acidity on the gut or different environmental conditions. So those things are things that one should take into account. Actually, for example, the germ-free mice, as you can imagine, to be born and raised without bacteria perhaps change so many things inside the mice that already the phenotypic change that you see when you give them a bacteria are not that surprising, right? But they are just consequence of the fact that they have never been in contact with any bacteria. For the human studies, then I will go a little bit faster. But for the human studies, let's say that we have a study since 2019, and it is important for you to take into account that at least on the bone field, most of them are coming, or I think up to now, the association, the observational studies, they come just from 16S technology, meaning that we are sequencing just the 16S, and then based on that, we are making a lot of assumptions to say, okay, this 16S gene belong to this bacteria, not to this bacteria, and then this bacteria has these genes and not other genes. So there are a lot of assumptions on all our analysis, but let's say that even at the population level with sample sizes quite small, they have seen differences between osteoporotic or normal BMD individuals. What I can say is that there is not reproducibility across the different small studies. So it's not like they have all point out to bifidobacterium or to Clostridium, or no, the results are all over the place. Also, it's important to notice that they use different type of adjustments when they do this type of studies. And of course, all these variables will, at the end of the day, take consequence on, the results that are obtained. Perhaps,
Carolina, we really need to move to the questions. We only have like 5 more minutes, so maybe can we go to the critical results and then
sure. So it is important for you just to consider that, that from the new studies, let's say that up to now, the largest study was published this year in JBMR. It was based on 1,329 men, and they look at different parameters of bone. But actually, I think the main conclusion from this study is that we need larger cohort sizes, because otherwise we will have still this problem with trying to reproduce results that are not really robust. So the issues that we have currently with the analytical pipelines is, as I mentioned, that when you are just sequencing one small gene from the bacteria, it's really difficult to make assumptions to which bacteria it belongs to and which other genes were present, because we have to remember that we are talking about bacteria. So there is also horizontal transfer from genes from one bacteria to the other that we are totally not taking into account. Also, that when we do these taxonomies, it can be that we are actually grouping things that are not that close to each other. For example, if I have the group in the left, and I say, okay, I will put together those animals that are able to fly because they should be closely related. I will finish with this group that perhaps, I mean, I have 2 birds, but I have also a mammal. So they are really different between each other. The problem with resolution makes that we are able just to get one species. And as I mentioned, perhaps the different strains will really make a difference if we want to translate our findings into the clinical setting. And that there are several ways in which we can approach the data. And actually, what we can see, we have seen in the last years is that there are all these papers that are saying, hey, look, for example, I took these 38 data sets and I analyzed them with all the different software that are available and used in microbiome research, and not one time I obtained the same results. So we know that all the pipelines are there, but we need to somehow harmonize them, because if everyone is using a different pipeline, we will never be able to replicate our findings. The reverse causality is another issue, meaning that we never know if it is the microbiome, the one that is affecting, like, as I mentioned, like, is it the microbiome, the one that is affecting the inflammation? Is the estrogen the one that is affecting the microbiome, and then the microbiome is affecting the inflammation? Is the estrogen playing a role in inflammation, and inflammation changing the microbiome environment? We still have to see how can we disentangle those relations. And of course, we need replication, but replication also implies that we have to take into account the heterogeneity, in the sense that everyone used different DNA extraction mechanisms or different pipelines for the processing and sequencing. And we know that at the end of the day, those are also things that will play a role on what we see and we don't see on the microbiome results. So the last 2 minutes will come from the other source of evidence that we have, that are the clinical trials that have been run. This is a paper from BMJ that actually, it was really nice because they summarized the ones that have been published. So we have up to date 5 different clinical trials that have finished and have published their results based on probiotics, and all of them actually have found differences, saying, hey, we gave these probiotics to people, and actually the BMD seem, because in this case they are looking at BMD loss, so it seems that the loss was less, not that they necessarily gained some BMD, but at least they didn't lose as much as the controls. So this is for lumbar spine BMD, but actually you can see results for other traits also on those papers. But still, our clinical trials up to date are not perfect. We know, for example, that many of those papers show sex-specific responses. So the success seems to be different between males and females. We know that the time and duration of the administration of the probiotic is an issue. So, for example, they all had a design in which they will give for 1 year the probiotic and then measure immediately the bone outcomes. But what happened if I took...
We have the time and duration of the webinar also, and we're approaching 1 minute, so we really need to go to the last slide.
This is the previous slide. This is the last.
So thanks a lot.
The take-home messages are that there is evidence. I know that there is these conflicting results, and that I'm really critical on how the field is moving, but it comes from diverse sources, so it's fine. We will find out more about mechanisms in the coming years, but there is a need on the harmonization of the pipelines for all type of analysis, and that we are more critical on the approach and the methodologies that are used on the field, and collaboration is key. So now